So before I begin with the scoring, I would first like to thank the challenge organizers for giving me the opportunity to work on the implementation on the scoring and playing around with the scores at the same time for the past 2 months. So this has been a very interesting challenge because in contrast to other challenges, the ground truth was actually given to the competitors up front. But the scores that were used to score the submissions of the participants were actually not revealed until today. So I will be talking about this one challenge, which is composed of these 2 parts, and for which we devised 3 scores to actually score the submissions. So the challenge is, in a way, was organized in 2 parts. So the first part was to find an optimal lower dimensional signature for each location, which will uniquely identify the location of each cell. And this was the task of reducing the dimensionality of the mapping from the 84 in situ genes that were used in the original paper into 60, 40, or 20 dimensions in the 3 different sub-challenges. So some of the competitors saw this as mainly a feature selection problem. And then the second part is actually given a function which maps the signature to a unique location to find a location-specific signature for each cell using only the transcriptomics data. Alternatively, because we know that there is also a mapping from each cell location into a 3-dimensional space, you can also find the 3D location for each cell from the transcriptomics data. And this is what is typically called in machine learning multi-target classification or multi-target regression problem for the alternative. So I was planning on going through and showing some theoretical limits about this challenge, but in the interest of time, I'm going to skip them. And I will just go to the conclusions and say that it is theoretically possible to find a unique signature for each cell using 60, 40, and even 20 signature genes. Actually, theoretically, this should be possible with only 13 signature genes. So these are the 3 scoring functions that were used in the challenge. So they're conveniently named S1, S2, and S3. So they focus on the 2 different parts of this challenge. So the first scoring function, S1, is answering the question, how accurate is the prediction of the most probable location? So maybe I should mention that the participants were required to provide an ordered list of 10 most probable locations for each cell. So S1, the scoring metric S1, was focusing on the predicted locations, but at the same time also on the subset of selected genes. The scoring function 2, S2, was answering the question, how accurate is the mapping function in general? And it used just the predicted location. And the scoring function 3 was answering the question, how accurate are the gene-wise spatial patterns given the predicted location? So it focused more on the selected genes and maybe somewhat less on the actual predicted locations. So I have to also note that from the 1,297 cells that were given, we had to remove 287 cells because in the original work, they were assigned somewhat ambiguous locations because the matrix correlation coefficients for them were same for several most probable locations. So the scoring was performed on the remaining 1,000 plus cells. So I'm going to show significantly more equations than the previous presentations today, but they are quite simple. So what is most important, can you see the cursor? Yes. We know that for each location, there are 3 coordinates, X, Y, and Z. So if we take, for each cell, we can take the submitted 10 most probable locations and just calculate the Euclidean distance between the submitted locations and the actual most probable location as was reported in the ground truth. So this is what this variable dk, the mean of dk, represents. So what this decay shows is actually the dispersion of the predicted location around the true location. And this is very very location-specific metric. So from this metric, we derived this ratio pk, which represents the ratio between the dispersion of the locations given the 84 in situ genes, so from the original paper, and the decay mean of the submission. And using this pk variable, we defined the metrics S1 and S2. So the metric S1 calculated the matrix correlation coefficient between the in situ descriptions of the most probable location as shown in the ground truth and the most probable location predicted by the competitors. And this was weighted by the normalized value of the pk for each cell. So the S2 is it's more simple. It's actually just the mean of the pk values. So, and for the third metric, so if we now compared, if you imagine spatially, we had in each row the signatures for every location, and we were comparing these locations using the matrix correlation coefficient. Now we are looking at columns. So we are taking the transcription data of the predicted location and the transcription and the in situ information for these predicted locations, and we are calculating the matrix correlation coefficient between them. So this tells us, if we look at this visually, if you can imagine for each gene, we can say that if the gene is expressed, we will color this with black. If the gene is not expressed, we'll color this with white, and we are just superimposing these two pictures and calculating the correlations between them. Of course, this is normalized by the values of the same metric using the ground truth. So those were the three metrics, and instead of using one or the other, we decided to use all of them to score each submission. So during the leaderboard phase, the competitors were allowed to test the scoring system, and this allowed us to actually also test the scoring system from the other side. So here I show the top 10 ranked teams. So for the different sub-challenges, we got a different number of submissions. So for the sub-challenge 1, we got 37 submissions, sub-challenge 2, 38 submissions, and sub-challenge 3, 40 submissions. Some of the submissions came from individuals, some of the submissions came from teams, but I will refer to all of them as teams, even though some of them are individuals. So in order to rank them, we first rank each participant according to each score, and then we calculated the average of these ranks, and then we re-ranked the participants using their average rank. And this is the final rank that can be seen in this column. But because some of the scores were relatively close, we decided for the final phase, for the final round, to actually adopt a bootstrapping approach, so we can be really sure that the winner of this challenge is significantly better than the other competitors. So we generated 1,000 bootstraps, and we repeated the same procedure that we used for the leaderboard phase for each one of the bootstraps, which left us with this table. So here we have the rank for each team for each bootstrap. This allowed us to generate such lovely box plots as you see them here. So this is the distribution of the rank for each team. And in order to assess the significance of the difference between the different teams, we calculated the bias factor. And this bolded one here is actually the indicator function, which this equation here tells us how many times for the 1,000 bootstraps the first team was better ranked than the second team over the opposite case. And in the box plots that you will see during when Pablo is going to announce the winners, you will see this thick black line, which actually separates the winners from the other competitors. And this line is drawn where the bias factor between the two consecutive teams is larger than 3. So this is all about the scoring. So we now move to announcing the best performance.
Okay, thanks a lot, Jovan, for really being so helpful for this. I had so much to deal with this challenge that, I felt you were there with me all the time. So thanks for that. Before just mentioning the best performers and seeing the ranks, by the way, they will be published Monday. They will be online, and you will have, all the data and the score functions and everything, so that you can check that we didn't do mistakes, I hope. But so there were a couple of problems in the challenge, and it's always good to say. In the feature selection, I think some people, it was not completely clear that, of course, you couldn't do a complete and free feature selection on 84 in situs, because the idea of the challenge was, well, let's say we have a new organism and we have to choose some in situs that we want to do experiments on in order to find the position of the cells. And so we had to make the mental exercise of thinking that we don't have the actual 84 in situs for the genes. So unfortunately, at least one important team didn't realize this and had to be disqualified, because this has to be fair for all the participants. Still, I think it's interesting to see the differences that would happen, and so they are actually presenting a poster, so you can see them a bit later. Then the second thing that happened is that because this challenge was a bit different, this is really, I think, the first time I have an open gold standard. The community has to auto-regulate itself, right? So it would have been very easy to just cheat and give the positions that were already available. In 99.9% of the cases, I think people were very good at doing this. So they really understood that the point was really having a performing algorithm that could predict, that could be universal. So, just not do overfitting, just based on the, up to 4,000 features that it had per cell. It was very easy to find a single signature for each cell. But there was a team that actually was best performer, and that we had to disqualify because the student's advisor realized that he cheated. So this happens, right? And I'm not going to say the name. I mean, you'll see the two disqualified teams, at least that were ranked top. Luckily, the advisor realized and emailed us right away. But these are things that happen, right? And I think I want to share the anecdote because it's interesting. It was pretty stressing for us and we thank all the participants of the challenge that are here today, because the other thing that was also very different was that it was a very aggressive timeline for the challenge, but we thought that because it was so interesting, it was really worth doing it. So we thank a lot people for being here, for having sent so many solutions. Okay, that's my speech. Okay, so here's the best performance for Subchallenge 1. The other thing that's really interesting is that Hung Goyen is actually the leader of Wata team. So they sent two solutions, but his solution was really orthogonal to what the team was doing, his students were doing. So I felt it was fine. And also they were, both top performers. So again, this will be published soon. You see that basically it seems that the advisor did better than the students, but they are actually really close, so it's okay. And actually, you'll see the two approaches. I actually personally think that the students' approach is more interesting, so you'll see. So for Subchallenge 2, yes. Well, the student is not here, but congratulations. Anyway, I'll just call them. Maybe if you can come here, Hung Nguyen, are you here? You should be here. Please come to the stage. We'll give you a best performing diploma. And then for the Subchallenge 2, so what the team also performed well, and this is something we see across Dream Challenges. Very often teams that perform well in a Subchallenge perform well in another. And then there's a second team, which is Omics Engineering, that is basically they're tied as best performers. And for the Subchallenge 3, again, Omics Engineering, which please, if you can come to the stage, I think they should be here, April, and the rest of the team, if you can come. And I can share again these with you guys, at the poster session. You will be able to see, you can ask me, I can show you on my phone the ranks again. So, and it will be made public. Okay, so without any views. Oh, sorry. Actually, also I would like to ask Thank You from Team MLB, if he can also come to the stage. He is not on the top 3 10 performers, but from the available people that come to the conference, he was the next one. So that's why we're very happy. And actually, he was very helpful during the challenge. So thanks for being here. Okay, so I want to give the diploma to the team Omics Engineering.
Thank you.
Congratulations. Congratulations. And Team MLB, I guess we're going to take a picture. Jovan?
You generated the data. It's your challenge.
So let's move this again, right?
Yes. I don't know why they did that.
No, you can sit down.
Wait.
How many? Thank you.
So you can stay, I guess, for your presentation. Okay, so now we're going to go into the presentation. Oh, I want to thank everybody, sorry, just from the challenges. You've heard their names, so